recorded, and if you miss any, you could always find them on our YouTube channel. In conjunction with the Brain Tumor Symposium on Wednesdays, which happens the same time every week, we also have a cerebrovascular and skull-based symposium that happens on Thursdays from 5 to 7. And tomorrow we have a great set of speakers talking about pituitary adenomas with Dr. Oyusiku and Dr. Velutini, talking about technique, results, and lessons learned. I'm sure this is going to be a great symposium with some great panelists, so be sure to tune in tomorrow night for that as well. And a quick teaser for next week, we'll be joined from the United Kingdom from Dr. Semendoras to talk about Journey to the Center of the Brain on pineal and third ventricular tumors. So please be sure to join us again next week. Some housekeeping, all participants, we try to make this as interactive as possible. After Dr. Stupp is done talking tonight, we'll be trying to get to everybody's questions. So please use the Q&A button at the bottom of your screen, and we'll talk to him about those. Also, we don't offer any CME, but you will get an email confirming your participation. And please be sure to like, follow, and share our videos on social media so that we continue to grow and share the fantastic knowledge of all of our speakers. So this week, we have a great set of panelists, starting with Dr. Plakantonakis, joining us from NYU, Associate Professor of Neurosurgery, member of the Laura and Isaac Perlmutter Cancer Center and the Kimmel Center for Stem Cell Biology. He's one of the investigators in the Neuroscience Institute and director of the Embryonic Stem Cell Research Oversight Committee, as well as the Neurosurgical Laboratory for Stem Cell Research. Dr. Prieto, who's one of our neuro-oncologists here at the University of Miami in the Sylvester Comprehensive Cancer Center, part of our neuro-oncology and neuroimmunology program. And I'll be filling in for Dr. Zachariah, who had a last-minute case and couldn't join us, and you guys know who I am. This week, it's really my honor to introduce Dr. Stupp. He really doesn't need an introduction. He's probably the most world-renowned and recognized neuro-oncologist in the world. He's currently at Northwestern, where he acts as the director of the brain tumor program. He's the chief of neuro-oncology in the Department of Neurosurgery. He's the Paul Busey Professor of Neurosurgery. He has titles in neurosurgery, medicine, and neurology. He's also the associate director for strategic initiatives at the Robert H. Lurie Comprehensive Cancer Center. Prior to joining Northwestern, Dr. Stupp was a professor in oncology at the University of Zurich in Switzerland. Since 2006, he's served on the board of European Organization for Research and Treatment of Cancer, or the EORTC. And in 2012 to 2017, he was the president. He also was a section editor during this time of the European Journal of Cancer. He has too many honors and awards and publications to kind of talk about all of them today, but I will mention he did receive the European Society of Medical Oncology Hamilton Farley Award and the Society for Neuro-Oncology Victor Levin Award, two of the most prestigious awards for neuro-oncology. He currently is active and dedicated researcher for both primary and secondary malignancies at Northwestern, and he's a key part of the Northwestern Brain Tumor Spore Program, which is one of only 11 in the country. And you can't talk about Dr. Stupp without talking about his landmark contributions for glioblastoma. Of course, his paper in 2005 here in New England Journal of Medicine, talking about the chemotherapy and radiation treatment plan for glioblastoma. And then more recently with his paper on tumor treating fields for glioblastoma, really the only two FDA-approved treatments for glioblastoma that we use and have made such a difference in the field and such an icon in this disease. So I'm just so honored and privileged to introduce him today and feel very lucky that he gets to talk to us about glioblastoma and his thoughts on this topic. So Dr. Stupp, thank you so much for joining us.
Thank you so much, Ivan, for this very kind words of introduction. I hardly recognize myself, and I apologize for the many titles. I'm not sure, is already sharing my screen? I don't think so.
No, if you could just reshare it, that'd be great.
Let's see, I need to get back to that. I thought by now we have it all figured out, but it's...
Okay,
that should be good. Okay.
Perfect.
So it's a pleasure to be among you. Of course, I think 2 or 3 years ago, I was down in Miami. That was more fun when you could directly interact. Can't even say interrupt. But please write down the questions so we can discuss. These are the obligatory disclosures, although I don't think I have anything that is really relevant to today. Very briefly, what are the challenges when we talk on glioma? And I'm going to focus on glioma, and I cannot talk on every one of the challenges, but I will really address very briefly that we have to do with infiltrative disease, the migratory potential, and the blood-brain barrier, and how to overcome those. On migration and proliferation, this is not really new. And I think that's what I was alluding to in my title. Everything is new and not so new. So this is work we know, on the left side, seminal works by Walter Dandy and others going with very aggressive hemispherectomy and so on, and not being able to really cure the disease. And then a very good review article by Giese and Björkvik in 2003 in the JCO, what it means in brain tumors and this migratory potential. This just to say that with surgery alone, or local treatment alone, we will not be able to have a full curative impact on this disease. To illustrate that in work that Craig Horbinski has been piloting, and really something we're not doing so much anymore these days, is doing autopsies. And this is out of an autopsy series, just showing that we actually see in a great number of patients when we do autopsy that we don't see anything necessarily on the MRI, but we see brainstem infiltration. So the tumor cells have to go there, and probably the brainstem is purely by gravity that the cells move along gravity. Maybe there are also specific pathways. These are things to be alluded to and worked on in the future. We've done trials trying to inhibit migration, but those have failed. And here, namely, like selengitide, that was the idea behind. Then surgery, and I'm not going to belabor surgery with all those renowned and expert surgeons among us. As a disclaimer, I'm just a simple medical oncologist, and you have better technologies. You have fluorescence, you have 5-ALA to do better surgeries, but this alone will also will improve somewhat outcome, but it's part of a global strategy, and that's what I want to get to. Nevertheless, yes, surgery, and this is the most recent paper out of UCSF showing the value of surgery. Of course, there will never be a randomized trial on the extent of surgery, yes or no, but this is very valuable because they really looked at it with the eyes of modern classification. And I come into modern classification just on the next slide: IDH mutation, MGMT methylation, and all what really matters very much so in the natural history. So not all glioblastoma, not all tumors are alike, even if they have and carry the same diagnosis. In brief on pathology, what I was alluding to, what you see on the left upper corner is some of the mutations we knew and how the classification would go with primary and secondary and grade 2, 3, and 4. And now I think most important when we talk about glioma, is it IDH wild type or is it IDH mutated? All oligos are IDH mutated, and then we have the diffuse astrocytoma with an IDH mutation, but also secondary GBMs that have an IDH mutation, while we still have the grading between grade 3 anaplastic astrocytoma and grade 4 GBM, but this has become much less relevant. MGMT as a predictive marker for response to alkylating agent, I'm not going to talk about today. You know that this has been there since 2005, and slowly we accept it also to take it into strategic decisions when we want to withhold treatment with temozolomide for unmethylated patients in favor of novel strategies. But what I want to just highlight that they are, when you have an anaplastic astrocytoma grade 3 tumor, if it has EGFR amplification, or it has chromosome 7 gain and chromosome 10 loss, or a TERT mutation, these are tumors that should be considered like a glioblastoma, and they are now to be called diffuse astrocytic glioma with molecular features of glioblastoma. This is out of work that is done by a panel of expert neuropathologists, but together with clinicians and others, to really try to see how we make progress in between the WHO classification updates. And it's called C-Impact Now Consortium for to inform on molecular and practical approaches to CNS taxonomy. And the now means, yes, of course, we want to do it now and not wait for the WHO, but it's not an official WHO classification yet. Then when we talk about radiation, which is another mainstay modality, just to highlight, we talk about radiation, we compare among trials, we talk about dosing, but across the globe, the radiation guidelines are not exactly the same. European guidelines are different than the US guidelines. The US guidelines are larger fields of radiation, but when at least we looked at it in trials we have done across the Atlantic Ocean between European patients and US patients, there was no difference in outcome. So again, we couldn't belabor that for a long time. I think even within the US, there are big differences. I know of US centers who go more by the more restrictive European guidelines. Of course, the size of the radiation field, the location plays a role. And when you look at clinical trials, it's actually shocking how little is reported, including my own trial, on tumor location or even tumor size. Not that the size probably by itself matters that much, but it matters with the location on the radiation field, on cognitive consequences of treatment, on late toxicity. And hopefully, as we get better with treatment, this will matter more. New radiation techniques. So we have gone from standard cobalt radiation, whole brain radiation, to focal radiation, to IMRT, and now more and more, and I know you have it down in Miami, proton beam, pencil beam, even more precise radiation. But does it really help in the field of malignant glioma remains to be determined. As I mentioned, it is an infiltrative disease, so the high precision may or may not be needed. In lower grade glioma, there is at least hints that you would have better outcome, less cognitive decline. That this is one series out of the Harvard group, MGH, with a small number of patients. They followed over 4 or 5 years. Again, the median follow-up is fairly short, and felt that they had over 5 years cognitively stable situation. Again, in favorable tumors. And I think here there is something surely to look into. And I think especially in lower grade glioma, the long-term survival and the cognitive decline is an issue we have to address, a thing we did not really worry that much 20 years ago. NRG, so that's former RTOG, now called NRG, is running randomized trials. And there is 2 trials I just want to highlight with protons. One is exactly in these low-grade glioma, BN005, looking at randomizing photon radiation versus proton radiation, followed by temozolomide afterwards in both arms. And primary endpoint is really cognition with a complex score of numerous cognitive tests that need to be done prospectively. Now, it's a complex trial, again, to do it multicentric with a complicated panel, an array of cognitive tests. take some logistics and again, collaboration, but I think it's going to be a very important trial. Now, the challenge is this trial to really mature will take many, many years. So we have to make sure that we follow these patients carefully after treatment is over, not only for survival, but also for cognitive decline. The other idea with protons is dose escalation. Does it allow to give higher doses? Overcome that? Now, this has been looked at, and so far, dose escalation has not really panned out in prior trials. But it was felt that dose escalation may have maybe some advantage, but again, all uncontrolled series. This is a series out of University of Michigan with Christine Etienne as first author when she was still there. You don't even have molecular markers. It's very hard to make a conclusion. So one trial we're going into is, again, with NRG, BN001, looking at the dose escalation. You see a very large trial, 567 patients. It's posted as a randomized phase 2. I haven't looked up the detail of the protocol, why it is a randomized phase 2. And it has a reasonable statistics of going for a hazard ratio of 0.72. Just as a reminder, I think a hazard ratio of 0.7 is what you can hope to achieve. Everything more is a bonus. For temozolomide, we had at the end a hazard ratio of 0.63, and also for tumor treating fields, we had a hazard ratio of 0.63. But so I think the statistics make sense. I think it's well designed. Together with temozolomide, we'll see what is going to be happening. The slide I just jumped before was just a general reminder on pseudoprogression. This has been reported sometimes more frequent with protons. Whether this is true in this case or not remains to be determined. But just remember, and this is even beyond 3 months, pseudoprogression remains a challenge when you do all the imaging. And so give the patient the benefit of the doubt if the patient is doing well, and treat beyond your radiological progression if the general condition of the patient allows. And this is important. I think most impact we have when we treat early on with the right thing, while actually second-line treatments have never really provided a good effect. So you don't withhold an active salvage treatment from somebody by treating a little longer. Then comes the blood-brain barrier. And this just a couple of examples. This was a young lady showing some contrast-enhancing tumor. But when you look at the T2, you see all of this is tumor. This is done at the same time. It's the same images. So actually, there is way more tumor extension than what we see. Even when we have more contrast enhancing, we can assume, and we have to assume, that there is tumor beyond what we see. And I think that's where the challenge is. That's where in the Molinaro's slide at the beginning, saying that actually supratotal resection, but even supratotal resection could never take care of this kind of extensive invasion. And sometimes you just don't see it, but it's still there. So the blood-brain barrier may be one of the reasons that many of the drugs didn't work. And if I take a series, and my list of negative trials is getting longer and longer, but all these drugs, including vincristine, selengitide, nimotuzumab, or afatinib, these were agents we knew going into the trials that they do not sufficiently cross the blood-brain barrier. And most recently, Depatax-M, ABT414, an anti-EGFR cytotoxic monoclonal antibody. We also know it's not crossing. Nevertheless, we have taken those agents into large randomized trials, and maybe we need to ask ourselves, is that really the right thing? So I cannot say that an EGFR inhibitor is not working in brain tumors, because if it never hits the target, then actually we will never be able to make any conclusion whether the pathway is not right. So how could we overcome the blood-brain barrier? There have been ideas a long time ago, like gliadel wafers, who would release cytotoxic agent within the brain. This was BCNU, but probably both the concentration of the carmustine or BCNU was too low, and also the penetration is not far enough. Then some of you have said, okay, we as neurosurgeons can take care of that. We do it with convection-enhanced delivery. There is quite some challenge there between you need a higher pressure. How do you get it really in there and so on? What is the right catheter today has also not really panned out. Implantation of radioactive seeds is something that is coming back, brachytherapy. But again, the extent of the X-ray without causing toxicity is probably a limiting factor. And then we have been medical opening of the blood-brain barrier or reversing the P-glycoprotein export pumps in the tumor. So sometimes you need to think a little bit outside the box. So here you have different ideas I want to address with you. So one was, can we use ultrasound? And there is different ways. There's a very focused ultrasound. This is work with coming from Toronto, using an ultrasound machine with an MRI, high intensity, very focused ultrasound. So produced by Insightec, that could help for certain neurological disease. But this high precision will also not really address the question we have in the brain unless we can have that to a larger field. Or what Alexandre Carpentier, innovative neurosurgeon out of the Pitié-Salpêtrière in Paris, came up with, can I implant an ultrasound emitter? And can I access the ultrasound emitter from the outside? So what was done initially, just implanting one single ultrasound emitter and then doing clinical trial with that. But one single ultrasound emitter is also too small of a field. But once this has been shown that this is safe and feasible, now we have a device that has 9 ultrasound emitters that could be implanted. Now it's invasive because it requires surgery. This will stay, this will be in a defect in the skull we have placed. That's how it would look like in the model. When you have the tumor, you would do it at the time you resect the tumor. It will nicely cover up. You will access the device with a transdermal needle that would go in here, that is then connected to the pulse generator. And in order to really open the blood-brain barrier, it's not only the ultrasound itself, it's the ultrasound in association at the same time of an IV administration of microbubbles that will then, through the ultrasound, being activated and mechanically open up the blood-brain barrier. So you need to have the right dose, the right frequency, the right duration, so you don't do vascular damage. And if you open the blood-brain barrier temporarily, so the protective effect of the blood-brain barrier is restored within 6, 8, or 10 hours. That's how it looks like intraoperatively. This is one of our patients. So Adam Sonam is the surgeon. So instead of the bone, we implant that. That will be then covered under the skin, but you could palpate it with your fingers, similar as you would do for a Portacat device. And just to show you a couple of examples, this is a young patient who had a recurrent IDH mutated tumor. A tumor was growing. This been the baseline after resection of the tumor and implantation of the SonoCloud 9 device. And this has been about 30 minutes after sonication. While at baseline, you don't see any contrast enhancement, you start seeing gadolinium enhancement shortly after sonication. and that's at the second sonication. And if you take this sagittal view, you can see that actually sonication field can be fairly large. So the opening of the blood-brain barrier to a reasonably large area. This is another example of a patient that's been before resection, after resection, and here again, about half an hour after sonication, you see opening the blood-brain barrier. If you do an MRI with gadolinium at that time, you see everything lighting up. If I repeat the MRI 48 or 72 hours afterwards, the gadolinium will disappear again. So that's all fine, but that's relative to what we have. We use the gadolinium as a relative measure of the blood-brain barrier opening. So how much does it open the blood-brain barrier, and how big of molecules over what time will really disseminate remains to be proven. So we have some gaps in knowledge, and I think one way is also to quantify, and we try to address that in a clinical trial I'm talking about in a moment, where really at the time we would do a sonication intraoperatively to an area that is non-enhancing, that has to come out in order to get to the enhancing tumor. And we would look and specifically biopsy there that tissue to then measure drug combination. Now for the ongoing trial, it has been carboplatin. For our upcoming trial, we purposely use Abraxane. This is albumin-bound paclitaxel or albumin-bound Taxol. So a drug that is potentially toxic, that clearly is too large to normally cross the blood-brain barrier, and try to assess how much we get there to then have a quantitative idea whether such an approach would really allow to study molecules that normally would not cross, or even trap molecules. Safety, of course, is a concern. So this is going to be a phase 1 trial. We start such a trial in recurrent disease, just as I have shown you, with the idea that ultimately at some point when we have shown safety and feasibility to use it in the upfront setting, at least for some patients. So you would get consent. We would have intraoperative sonication only with one sonicator, look at biological endpoints, at tumor changes of the tissue, and genetic changes, upregulation of certain genes, and drug concentration. The beauty about this concept is that you can do it repeatedly. With that implanted, we could afterwards non-invasively, through this, with this transdermal needle, access the ultrasound device and treating in cycles the way we have established it in oncology for many diseases. And we have established actually for GBM also with temozolomide. So this trial has just been approved, is activated at Northwestern, or is about to be activated in the next week or so, so for the first patients to be treated. What are we going to compare it with? It's going to be a non-controlled trial. It's a phase 1 with an expansion cohort. So we need to look and compare it. At some point, we also want to know efficacy, not only safety. So what we will compare it to some extent is the recently published TokaGen trial, or presented TokaGen trial, which actually showed there has been no benefit of TOCA 511 with repeat surgery. All those patients had repeat surgery. So this is a very good indicator what we can obtain in recurrent disease with best physician's choice chemotherapy following initially repeat surgery. So we get about 12 months. So we'll see what we get in our early phase trial to see whether this is a concept to take forward. You remember I said outside the box. So in the outside the box, what would come else? Tumor treating fields. I trust we have talked about it before, but I still want to show briefly where we're going and what this concept does. So tumor treating fields where we have various mechanisms of action. There is also at least some preclinical data that tumor treating fields will increase permeability of glioblastoma cells, and therefore probably works best when you have it in combination with chemotherapy rather than just alone. And it would allow for better penetration of the chemotherapy agents into the brain. And so this is a concept that has been advanced and work that has comes out of late Sanjeev Gambhir's lab. Chirag Patel, who was working in his lab and is now faculty at Stanford, will take this to the next level and further investigate that. You've seen how this works, how you practically do it. You'll see that the device has become smaller, and it can be really done. I think it's feasible even for ladies. This lady didn't want to take it off after 2 years. That's about the duration that was investigated in the study. Where do we go from here as next steps? The next step would be with the idea of combining it with radiation early on, because there is data not only for synergy with chemotherapy, but there is also preclinical data of potential synergy with radiation therapy to now evaluate it in the upfront setting. So already starting it during radiation. The reason at the time that the trial was started and randomization was after chemo RT was that it would have been impossible to convince the radiation oncologist at the time to irradiate with the electrodes in place. Second, to have the electrodes placed and taken off for 6 weeks in a row on every day, I think for the skin may not be the best solution. So that has been another consideration why this was not done at that time. And again, don't forget these trials were all designed 2006-07. So it's been over a decade. So now I think the company at least feels the time is ripe to start it in an upfront setting. And they have designed another large international trial with up to 1,000 patients to be randomized, looking for a difference in overall survival. Quite an ambitious trial, also an ambitious target or endpoint to improve overall survival just by starting 2 months earlier. So it will be interesting to see. My colleagues, Rachel Grossman and Tzvika Ram from Israel, from Bellinson, have shown that this can be done. So they have an ongoing trial. They have already included close to 60 patients looking at this, that is feasible. So that's ongoing, that's on the horizon. What else is on the horizon? PARP inhibitors have been used and have been a come and go and had somewhat of a renaissance. So basically interfering with the repair pathway in glioma with PARP inhibition. And this works. This works in ovarian cancer. This works in certain prostate cancer, in BRCA mutated tumors. So there has been some interest to combine that with temozolomide. And Carrie McDonald and her team in Australia have done quite some good preclinical work showing that there would make sense to actually add and investigate veliparib in unmethylated tumors. They have presented the results about a year ago at SNO, and as you can see, no difference in progression-free survival nor overall survival. Similarly, for the methylated ones, the Alliance Cooperative Group has taken this forward and has run a large randomized phase 3 trial looking at adding veliparib to temozolomide after chemoradiation during the maintenance phase. This trial has fully recruited and we are waiting for the results. So I think by this time next year, we should probably have the results. Because it's the methylated patients, you need way longer follow-up than in the unmethylated until you can conclude. What else is out there? Immunotherapy, and of course, it's just impossible these days to talk and give a talk on trends without mentioning immunotherapy. But is what is true for melanoma and lung cancer truly also applicable to glioblastoma? Glioblastoma is a completely different disease, has a low mutational burden. Smoking will cause a lot of mutations, so will melanoma with UV light. What about in glioblastoma? This is not the case. And indeed, a trial run through Bristol Myers Squibb with 360 patients comparing in recurrent disease, nivolumab versus bevacizumab, there was no difference in outcome. This has just been published finally in May this year. Why it took 3 years to publish that? If this would have been a positive trial, it would have been coming up at the day of presentation. That's beyond what I understand. Now, sounds like that somebody at Bristol Myers must have heard me before when I was advocating that you should, if you want efficacy, try to really move in the upfront setting, because even temozolomide or tumor treating fields in the recurrent setting has not shown to improve outcome. So they have been running in parallel 2 more trials, separate for methylated and for unmethylated patients, and looking at adding nivolumab to the standard of care. I don't have the survival curves, although they must be out there somewhere, but we have 2 press releases that are fully disappointing, showing that it has no effect whatsoever. So unfortunately, negative. That doesn't mean immunotherapy doesn't work, but probably unselected patients, single agent, may not be the right approach when it comes to diseases that have a complete different pathogenesis like glioblastoma. The other thing which is quite impressive, there is numerous trials with checkpoint inhibitors out there. Many of them uncontrolled variations on a theme, not sure they're going to inform us on that much. But when you go on what has been out there on checkpoint inhibitors in the literature, you see that the high number of patients are on corticosteroids. And I think if you want to go in immunotherapy, you need to remove the most immunosuppressive drug we have, the corticosteroids, and otherwise you're not going to see an effect. And I think even in the phase 3 trial I just shown you, there's been a high percentage of patients who have been on, sometimes we call low dose, but even 2 milligrams of steroids will cause lymphocytopenia, is somewhat immunosuppressive. So this should be off. If you want to combine it, at least I often combine it with low dose bevacizumab. Low dose means 3 to 5 milligrams per kilogram instead of the 10 that has been approved, and give it every 3 to 4 weeks. And it will allow you to have the great majority of patients off steroids. This just to remind us, whatever series we look at, patients on steroids have a worse outcome than patients without steroids. And this could be shown in the preclinical model. So it's not only selection, that's a mouse model coming out of Eric Holland's lab, showing that steroids also in the mouse are detrimental. Now, after all this negative propaganda on checkpoint inhibitors, we have nevertheless a positive trial. Now, this is an important Nature Medicine paper by Bob Prince and Tim Clousey and collaborators, where they randomized patients with recurrent disease to receiving neoadjuvant pembrolizumab versus surgery first and then the checkpoint inhibitor. And it allowed a lot of correlative science, changes in gene expression profile, and so on. What, however, got a lot of attention is their survival curve. Now, again, I showed you for BN001 or 005, they need 500 patients. I showed you our trials have always been several hundred patients. And here you have one arm with 19 patients and one arm with 16 patients. I think it's just not enough to really show a comparative survival curve. And this may just be patient selection. So that's, I would kind of caution. Nevertheless, it's very intriguing. I think the principle of looking neoadjuvant before you do repeat surgery or even before you do initial surgery, I think something to really keep in mind as a strategy to explore, to learn, and to ultimately improve outcome. What about oncolytic viruses? Another approach that has been investigated and is being investigated in patients in recurrent disease. The mode of action is variable, direct oncolytic viruses, but also presentation of tumor antigens, stimulation of immune response. So maybe best if combined with some checkpoint inhibitor and other immune modulators. So one example is this virus produced by a company called Xiopharm. It's an adenovirus that has been genetically modified with the Rio switch that would allow to turn on and off by giving validomex human IL-12 secretion. And when human IL-12 is secreted by the tumor, you get a lot of inflammation, you get a lot of signaling and attraction of immune cells. So basically trying to make a cold tumor hot. So this is what has been done. It has been a first trial with 31 patients showing that this is possible. We have a better report now by Nino Ciocca, came out in summer of last year, showing that you really can induce an immune response, that you have post-treatment a lot of positive immune cells. We have high excretion of secretion of IL-12. And when you have treated patients like this, you know that they had a lot of inflammation. Sometimes you could not give it every day, the validomex, because you even needed to slow it down. So the system per cell itself works. On survival, I think, again, in a phase 1 trial, very difficult to make any survival conclusion. But the next step, what we have done on this trial, and which is ongoing now or has completed recruitment and awaiting the analysis, has been this strategy combining with a checkpoint inhibitor. And that's also how things will go forward. Just to show you one patient on this, David is a 60-year-old gentleman with a left frontal lobe. He had a gross total resection in 2018, went on a clinical trial there. He had a non-methylated tumor, IDH wild type, so kind of your off-the-shelf poor prognosis glioblastoma. He had a recurrence 6-8 months later, and he had a second resection with us and followed with this adenovirus injection plus an immune checkpoint inhibitor. So just to show you the sequence of things, this has been in January, the recurrence before resection. It was hyperperfusing. It wasn't pseudoprogression. That was also confirmed pathologically afterwards. And this is 11 months further, but actually now we are 16 months post-treatment and 27 months post-diagnosis and not the sign of recurrence. So you would say, oh, this strategy works, and it may well work. But what I want to get, when we did the second recurrence, we did some more molecular analysis. And we actually identified that he has an FGFR3 TAX3 fusion. And these tumors have a different natural history, and they have an overall better prognosis. So that explains why this unmethylated tumor actually has a survival of over 2 years. So our treatment may have an effect, but we also have a different makeup. Also, just to warn, when we do things and we compare it, small numbers with uncontrolled, randomized, we need to know more details. Not all tumors are alike, no matter how they look under the microscope. What it also tells us that for this patient, should he once recur, that we would now have FGFR TAC inhibitors that are in clinical trials, and one has also been recently approved, not necessarily for glioma, but it is at least on the market. I think it has been approved for cholangiocarcinoma, so that's things, at least we get access to the agent. Now again, do the agents reach the brain in a sufficient concentration? I think that's something we need to address as we do trials in the future, that we do trials and we really look at non-contrast enhancing tissue to get a dosage to see whether we have really cytotoxic there. Another agent is larotrectinib for NTRK inhibitors that has shown some effect and got some leadway and interest in brain tumors with good responses. Again, these tumors are less than 5% of the glioma, but if you don't look for those aberrations, you will not find them. So let me sum up. I think we have plenty of modalities, but we need to work in concert and we need to work together. We need neuroradiology, we need advanced imaging, which I didn't talk about, PET and other ways. We need them in a team and we need them in a dialogue. Interdisciplinary collaboration, I think you have it in Miami, we have it in Chicago, but that makes the difference when we really want to offer the best possible care to the patient. Care means not only treatment, but also things in synergy, but also knowing when we need to have palliative medicine involved and when we can really address also the goals for the patient. I need to acknowledge lots of this work has been done with my partner in crime. This is a very talented young neurosurgeon, Adam Sonnabend, who here was wearing a mask when we were actually traveling the Middle East and seeing one of our patients in Qatar who was on one of our surgical trials before. Preclinical work that I didn't show you but has been published by Dan Zhang that shows all the preclinical work that was there in order to do the clinical trial with Abraxane and ultrasound that we now have up and going. The collaboration with neuropathology, we are spoiled that we have Dan Brad and Craig Robinski in the team, and Craig is even part of the Department of Neurosurgery on top of being of Department of Pathology. Denise Scholtens and her colleagues as statisticians. We have Chris Amidei, an outstanding research nurse with a PhD, understanding and running the show, helping us. David James with all the preclinical model, Matt Lesniak, Department Chair of Neurosurgery, outstanding. And then we have neuroradiology. We have people in collaboration helping for dosing, also in animal models. This is from Illinois Institute of Technology and our partners in industry. And of course, to acknowledge our funding the Malnati Brain Tumor Institute, which is the philanthropy behind our Brain Tumor Institute and our endeavor within the Lurie Cancer Center. And then also philanthropists who have helped to give us the freedom to move forward until we could get the grants, and with whom, without that support, many things could not have been done. Really want to acknowledge NCI, the SPORE grant we got, the R1 we got, And these are grants Adam Sonnabend got early on in his career. So that's the team it takes to make music in Chicago. If anybody wants to join us, I think we're talented people. We always have space. And otherwise, let's try also across the country to collaborate in order to really make progress in this disease. With that, I want to close. Thank you very much.
Thank you so much. That was an amazing talk on some of the latest and greatest and very inspiring. So let me just ask a few questions here that we have from the crowd and that have come in before we get to our panelists. Rachel Yan was asking specifically about the implanted focused ultrasound device and how does that compare over some of the other external devices and HIFU and whatnot. And is it something that you could use on recurrence, cost correlation and things like that? Any comments?
I think to talk about cost right now, it's way premature. This is research. I think the most costly treatment is all the treatments we do that don't work. So I think if some, the difference is that we don't have to overcome the skull. So it's much easier. It's a much simpler once it is implanted to do it repeatedly than the external. The HIFU with the InsightTech device is complex and it's very precise, which is great. But I think for precision, I have my neurosurgeons and I have my colleagues in radiation oncology who can also get the beam very precise. So I think it has its inherent limitations. It's not one or the other. I think it's both ways should move forward and we learn from each. I think the fact that we can now irradiate large portions of the brain is interesting. Not irradiate, sonicate. We are now at 9 ultrasound emitters. If the concept works, we could even have 18 ultrasound emitters. I think there would be ways until we have ultimately maybe something that we don't need to implant. But to my knowledge, and I'm not a physicist, to overcome with ultrasound to skull is quite challenging.
Great. Bruno Daluz asks, why do you think there's not more research and clinical trials focused on early identification of pre-tumors and understanding kind of why these tumors are happening in the first place with liquid biopsies and screening the general publication to do preventative cures rather than treating disease after recurrence?
So there are two things to it. A is the liquid biopsies is something that we and others do, and I didn't go into that in interest of time in parallel. I think, can we follow, also can we get a response on a liquid biopsy early on, what is happening, whether our treatments are effective. For prevention, similar as screening, that only makes sense when we have good treatments. And even there, prevention is highly disputed, even with even nowadays still with mammography, whether it helps, or PSA screening. So we could have day-long discussions on those. So I think as long I don't have a good treatment for glioblastoma, I'm not sure I want to come and see you, that you let me know that I have 2 years in advance already misery, knowing that I'm going to succumb to the disease.
Okay. See, Ernesto talks about transcranial magnetic stimulation in the role of recovery for glioblastoma. What are your thoughts on using this for identifying, eloquent areas and also for areas of recovery?
I think you guys are better positioned to answer that question than me. My disclaimer at the beginning, I'm just a simple oncologist. And I think, yes, we need to do more. What I alluded to at the beginning, that we don't even look at location in detail, despite all the neurologists we had in the team, I think is a big missed opportunity. And I think we do to do better in the future.
And one last question before we get there is is with all these clinical trials and that don't profile and look at the subtypes of tumors, do you think that's going to be the new requirement now? And if so, how do you know which profiles are important? It seems like every year we're coming out with new subtypes markers within them, and how do you keep track of them as you try to go from one clinical trial to the next in order to understand who's benefiting and who's not?
I think here the team comes in. Even I can't keep track of all the markers at all times. I think the discussion, there is even other issues. It is quality assurance. You always get a result. Is the result reliable? Is it not reliable? I think with MGMT, there was major work just for a single marker to validate it, and it was validated. We still fight on the technology. I could, again, I purposefully did not go into MGMT, but what you call methylated or unmethylated will depend, because there's a gray zone in between what you want to enrich for. So if you want to withhold temozolomide, you have to be more restrictive, what you call unmethylated, than if you want to enrich for methylated ones. So there's all kind of factors that play in. So it's not the same cutoff.
Okay, great. Yeah, there's a bunch of other questions, but I want to make sure we have time for our panelists. So Dimitris, you want to start with your case and talk? Just you want to unmute as well.
Okay. Can you guys hear me?
Yes. We're looking at
Okay.
Virgin perspective slide.
Okay, sorry.
There you go.
Okay, Mike, thanks so much for the opportunity to speak here. And what an amazing talk by Dr. Stupp. So it's hard to follow Dr. Stupp in this forum. But I'll do my best to tell you a little bit about what I do here at NYU. I'm a brain tumor surgeon at NYU. The review that was given by Dr. Stupp was really comprehensive and amazing. And congratulations, Dr. Stupp and my friend Adam Sonnabend on the innovative work with the ultrasound. A couple of disclosures, get that out of the way. So this is the Dr. Stupp's paper from 2005 on the concurrent use of temozolomide and radiation, which is the current standard of care for newly diagnosed glioblastoma. And it certainly made a difference. We've been using it here for almost 15 years now. But when I started working on glioblastoma, I looked back in older literature and I found this amazing paper from Montefiore Hospital, actually, here in the Bronx in New York. It goes back to 1949, so 70 years ago. The median survival for glioblastoma was 8 months, and now we're up to 16 or so. So we really haven't made very much progress. It's about an additional month of survival for every decade of research. So that underlies how tough this disease is. And I recently came across, I was doing some retrospective analysis of patients of mine for a study, and I came across 2 patients with an essentially identical tumor. And both of those tumors end up being IDH wild type MGMT methylated glioblastoma. So this is a 64-year-old gentleman who presented with gait imbalance, behavioral changes. He had a right temporal tumor, a large one, conducive to aggressive surgery. We take it out. As you see, the diagnosis was IDH wild type, MGMT methylated glioblastoma. And he got temozolomide and radiation. He's doing great 4 years later. Next month will be 4 years, actually. He has now he's developed in the past few months some subtle progression in the ependema of the right lateral ventricle, but clinically he's doing great. This is a tale of two tumors that look identical in MRI, but they're biologically very, very different. This is a 72-year-old female who presented with confusion, and the MRI showed a near identical tumor as the prior one in the right temporal lobe in an area conducive to aggressive surgery. And we get a gross total resection, and this is a more recent case where we made use of intraoperative MRI and 5-ALA at our institution. We're lucky to have these technologies. And we get a gross total resection, but despite her being IDH wild type and MGMT methylated, and therefore expected to respond very well to oculating agents, she was treated with radiation and temozolomide, but about 6 months later, she developed diffuse ependymal leptomeningeal spread of her tumor and passed away. So it's a tale of two tumors that look identical in MRI, and they're both expected to respond well to chemotherapy, but they behaved very, very differently, the one from the other. And it points out how little we understand about the biology of these tumors. So from my perspective, I know as a surgeon that no matter how good surgery is, and no matter how much technology we add to the surgery, we will never cure these patients with just surgery alone, as Dr. Stupp said. And unfortunately, we still don't understand the biology very well. As a surgeon, what I think about when I do surgery for glioblastoma is I try to achieve maximal safe cytoreduction. We take out as much tumor as we can safely, but we're aware that we can't cure anybody doing surgery alone, and I try to reduce mass effect. But as a scientist, I try to understand the biology of glioblastoma to try to make a difference in the care. So I want to show you a couple of slides about some of the work that my laboratory is doing. The past few years, we've been working on a class of proteins that we find very interesting for several reasons. But these proteins belong to the family of G protein coupled receptors. G protein coupled receptors represent one of the largest protein families with about 1,000 different members. And they are transmembrane proteins, and they couple to so-called G proteins that entrain signaling mechanisms within the cell. So these proteins transduce extracellular signals like ligands or stimuli, and initiate signaling mechanisms inside the cell. Within this family of proteins, there's a subfamily called adhesion G protein coupled receptors, and those adhesion G protein coupled receptors have a very long extracellular N-terminus. And the past few years, we came across one member of this adhesion G protein coupled receptor family called GPR133 that looks like this. This is a cartoon showing the membrane topology and domains of the protein. And we've become very interested in it. And one of the reasons, I'll show you a couple of the main reasons why we're interested in it. This is a putative structure of this protein. But GPR133 is not expressed in the normal brain, including areas where the ependymal area of the ventricular system where progenitor cells are located. I should say, related to one of the questions that came up earlier, these progenitor cells may be a cell of origin for glioblastoma. But when we studied the expression of this protein, GPR133, we see it come on more and more as gliomas go from grade 1 to grade 4. So it's expressed in all glioblastomas that we've looked at. Within glioblastoma, it's expressed in within the core of the tumor, but also in the infiltrative edge of the tumor where the tumor invades the brain. This is a cardinal biological behavior of glioblastoma that, as Dr. Stupp said, precludes surgical cure. The expression profile within the brain is favorable for targeting this protein. It's de novo expressed in these tumors, essentially. I'm trying to advance, but
click on the presentation. Click on the screen on the presentation and then advance.
Hold on a second. Trying to do that. Oh, here. Okay, and when we look functionally at what this protein does, there was very little known about this protein before we started our studies. We used the technology called knockdown, where we experimentally can drive down expression of the protein in the tumor cells. These are patient-derived tumor cells. So here you can see an experiment where in the control experiment, we put patient-derived glioblastoma cells in the mouse brain, and they form a tumor. The green cells are human glioblastoma cells growing in the mouse brain. But in the control, in the experimental condition where we bring down expression of this protein in the tumor cells, the human cells cannot form tumors anymore. And this is the mouse experiment summarized here. And the mice that have the control, the human cells that have expression of this protein, they die. But the mice implanted with cells that don't have this protein, they keep going because they don't form tumors. And this parallels what we see in humans. So when we take the Cancer Genome Atlas database of GBM patients and dichotomize the patients by expression of this protein, expression of the mRNA encoding this protein actually, we see that the high expressors in red live shorter than the low expressors in blue. So this suggests that this protein helps the tumor grow and perhaps be more aggressive. How do we use that information? And so in my laboratory, we do a lot of basic biology to try to understand how this protein works. But from the translational point of view, we tried a couple of years ago to generate small molecule inhibitors, and we were not successful in finding specific inhibitors. But right now, what we're trying to do is develop biologics. What does that mean? This is antibodies or related molecules such as monobodies, for example, that recognize the long extracellular domain of this protein, which is predicted to be highly antigenic. And we're collaborating with researchers here at my institution to develop either neutralizing antibodies or think about the concept of antibody drug conjugates. So an antibody that recognizes the protein and without having any specific function on the activity of the protein, it just carries a poison, a drug that kills the cell. And we've recently have become interested in a related protein called CD97 that seems to have a similar expression profile, not expressed in the brain and expressed in glioblastoma de novo. And that's a protein that was studied actually by the late Andy Parsa at UCSF and Northwestern in glioblastoma. So we're trying to further the work of Andy Parsa a little bit, and we think it may be interesting as much as GPR133. One of the other things we're doing in my laboratory is looking at one of the future directions of basic research that Dr. Stupp pointed out, the epigenome. The epigenome refers to not just changes in the DNA sequence, mutations, but changes that relate to proteins that bind the DNA or the organization, the three-dimensional organization of the genome that affect gene expression. These are some of the concepts that I just alluded to as epigenetic modifiers of gene expression, chromatin looping, three-dimensional nuclear organization, and enhancer promoter interactions that may underlie glioma initiation and progression, And I'm not gonna get into that because it becomes a little esoteric trying to explain it, but we have found that in the initiation of IDH mutant glioma, there are changes in how chromatin, the DNA of cells, loops. And this looping is essential to gene expression. Several genes that are crucial to oncogenesis in glioma depend on this appropriate chromatin looping. And where this chromatin looping is dysregulated, as happens in glioma initiation, certain genes are turned off and other genes are turned on. And that can promote the oncogenic transformation of a progenitor cell in the brain. Some of the analytic tools that we have to understand chromatin organization, chromatin looping are shown here. It's a little esoteric, so I'm not gonna try to explain it, but it looks kind of cool. It's nice to show. But these are some of the things we do in the laboratory. So we try to understand biology as much as possible, and we try to be thoughtful about what we take from the laboratory to try to bring to potential clinical trials. I'm going to stop here and thank my lab and our many collaborators, but also folks that help with our clinical work, and the folks at the Brain and Spine Tumor Center here at NYU.
Thanks so much. That was really great work that you guys are doing, and it's ironic because I was with Andy while he did that work at UCSF and CD97, and we're actually working on CD97 a little bit in my lab, so we should talk a little bit later.
Yeah, good, absolutely.
All right, perfect. Pilar, we could switch to some cases now. I know Pilar has some cases for Dr. Stupp.
Yes. Just, how do I do
Hope the cases are for all of us.
Yes, they are. You could just share your screen. If not.
Oh, okay. Hold on.
If not, I think I have your talk. Let's see.
Let's see.
Let me get it up.
I think I got it.
Okay.
In the meantime, for the ones I saw, there are a few more questions on in the chat. I just made a photo of the chat, so I'll try to reach out to you with Michael's help to find your emails and respond later.
Perfect. Thank you.
Hello, everybody. I am Maria del Pilar Guillermo Prieto Ivel, for short, Pilar Prieto. I am one of the neuro-oncologists here at the University of Miami. Thank you, Mike, for the invitation. And thank you, Dr. Stupp, for such a great talk and for all the contributions that you've made in the glioblastoma world. It's great to hear you talk and to actually meet the person that that has revolutionized the treatment for glioblastoma. So I am presenting an interesting case of H3K27M diffuse midline glioma. This is a 49-year-old Caucasian man with H3K27M diffuse midline glioma of the cervical thoracic spine. Onset of symptoms started in August 2019 with right arm weakness and pain, and he attributed it to injury of his arm while he was working. He's a contractor. MRI on January 3rd, 2020, he had a right hemichord expansile intramedullary infiltrating process, which we can see this large lesion, and had some patchy enhancement. He underwent biopsy, and the biopsy showed, was consistent with a diffuse midline glioma. He completed radiation therapy on the spine on April 5th, 2020. And then a month later, the MRI showed significant edema concerning of necrosis with probable progression. So you can see how it fills up the spinal canal. He was started, interestingly, despite how horrible his spinal cord looked, he was still ambulatory. He had mostly right-sided symptoms with spasticity and neuropathic pain. But throughout the summer, he was able to renovate his own bathroom and make it look like a magazine, like one of those like really pretty bathrooms accessible for handicap, in preparation of what may come later. So this is 4 weeks after radiation therapy. He was placed on high-dose steroids, and we were trying to get him on the compassionate use of ONC201. But just like around the time they closed, and the trial available for him here in Miami for ONC201 was temporarily closed for enrollment too. So he was started on bevacizumab on June 30th. So far he has received 4 doses. The last one was on September 9th. He also received 2 cycles of Temodar, and on August 7th, he had a cervical spine MRI which showed improved edema, and the enhancement was better, probably because of the Avastin. And at that time, he reported some vision changes, for which I ordered MRI. Then, later the patient said it was just dry eye. So, but I kept the MRI. Cervical spine on September 5th showed disease progression. It was a caudal extension from the tumor, from T1 to T3. The patient was mildly symptomatic from it, and just a little more spasticity and more difficulty moving the right leg, but not a significant change from his baseline. And based on these changes, I was able to get him in contact with the team for ONC201 here. These are the images from May 7th, and then to from August 7th, and then to September 5th. You can see how the tumor feels in the spinal canal, especially in this area here. So so then, in the meantime, he also got the MRI of the brain, and we can see that there's multiple new T2 flare lesions, in the corpus callosum, in the brainstem, in the cerebellum, and in the left temporal lobe, and in the medulla oblongata. So, and also there's like some cerebellar lesions that we're tracking in the in the cerebellum, like in this area here, look like possible leptomeningeal disease, and none of these lesions were enhancing, and these lesions are not symptomatic either. The next day he presented. This is the cervical spine. That was done, and it showed the stable changes from what was noticed before, and the involvement of the brainstem right here. On September 19th, he presented to the hospital with acute lower extremity weakness, urinary retention, and bowel incontinence. He had a lumbar spine MRI and showed a new lesion in T12, which is not enhancing. He was admitted, started on high-dose dexamethasone, and started emergent radiation therapy. On the exam, he has an old right Horner syndrome from May 2020. I would just like to mention, back in May, he also had a brain MRI, and the brain MRI was clean. On his exam now, the rest of the cranial nerves are intact. He has right upper extremity weakness and spasticity, which is stable from prior. But the new changes were that he had a bilateral lower extremity weakness, much worse on right and left, and had a new sensory level at T12, and in addition to the prior sensory level he had, and the new urinary and bowel symptoms. With high-dose steroids, he recovered a little bit of movement, and apparently today he was able to to give like a 4 out of 5 strength in effort to the examiner today, and because of the possible leptomeningeal disease, he's no longer eligible for ONC201. Now we did CSF analysis, which is pending for the cytology is pending, in the hopes that he may be able to be a candidate for this trial. So with this, with this challenging case and this challenging tumor itself, we know that ONC201 does not cross the blood-brain barrier, so there's an unclear benefit if he does have leptomeningeal spread of the disease. ONC206, it seems that it crosses the blood-brain barrier, so that might be a possibility, but it's not available yet. Lomustine alone or in combination with Avastin might be a possibility, or potential role of immunotherapy along with Avastin plus minus radiation to the brain. We're currently holding radiation to the brain until the patient becomes symptomatic, because of the concern of inflammation and necrosis, and then we accelerate the disease process and potentially be locked in. So with that having said that, with Dr. Stupp, with your experience, do you have any recommendations to treat this patient better? Even maybe a potential role for tumor treating fields for the intracranial disease?
I'm glad you're keeping it very simple, Pilar. This is a very challenging case.
So when Michael and Ricardo reached out to me whether I would give a seminar, of course, they didn't tell me that they're going to challenge me to that point. So I'm going to probably have to disappoint you. I think I agree with all what you have said. And the truth is, there is no good solution. So maybe just a few considerations. I think probably nothing will work. I'm not sure we have any data supporting temozolomide in K27M mutated tumors. I would probably also have given it just short of something better, but it's a concern, especially when you have sometimes less aggressive tumors and you combine it. And here you don't, for once, don't want to have too much inflammation or radiation necrosis. So that's just to be considered. I think CCNU will also, I think it's the next line to give, and it crosses the blood-brain barrier, but very unlikely that it will really help. ONC206 would be the right thing. Now, the difference between ONC206 and Temodar is ONC206, we don't yet know that it doesn't work, while for Temodar we do. So you work on the hope, that's why you have those investigational agents and clinical trials. And we need to find altogether ways that for patients like this, there is access to the drug one way or another. I think cost came up repeatedly, but what we're doing here, trying everything under the sun, uncontrolled, that's the costly part for this. An outside COVID patient could travel somewhere, so there must be ways. Now, the story for Oncocytics. This is a very small biotech. They don't have the resources to really provide the drug. We have the same issue that the drugs are not available in Europe at all without funding. So this we have to see, and we have to run the trials with less overhead. The thing you mentioned immunotherapy, always comes up. I think if you want to do more and there is the strive for more, that's probably of the unknown, which has probably the longest potential benefit. I would probably then combine it and give immunotherapy before I give radiation to the brain, just for this idea of abscopal effect. Now, it's always talked about, this abscopal effect. It's always the same people who have seen it. I'm still waiting to see it. I've given years ago radiation plus checkpoint inhibitor and even Avastin to avoid the steroids and re-radiation. all of that, I couldn't pan out an effect, but at least in the lab it works, and there are individual case reports. Now, if you have to be careful with the resources, I think probably the best resource is at the end in palliative care, and less is more. I remember it was young, but I don't remember the age of the patient. But
49.
how much?
49.
49. But of course, people don't want to hear that. But I think early on to have discussion what matters to their life, what is important. It's also what is important that the family doesn't end up with medical bills that will have them suffer for the next 20 years. So all these kind of considerations, but a good solution, I don't have, you mentioned it all. So it's all kind of considerations.
Dr. Stupp, there's like just a preclinical study of alternating electric fields in vitro for diffuse glioma cells. I mean, diffuse spontaneous glioma cells, and it seems that there's like some positive effect from tumor treating fields in stopping these cells from growing. Do you potentially see a use for Optune in this case?
So you could, Novocure tells me that they have now ways even for pontine infrastructural location. Now again, you have disseminated disease. So you're going to treat where the fire is the highest. The effect, it takes usually months. So I would not have come up with, I would not be against. I'm sure Novocure will support you as much as they can. But again, how much do we really gain when you can't cover the whole field with the spine? That's why I didn't put it high on my list. Yeah. If I take the data on tumor treating fields now outside this K27M mutation, which I think, at least from what we know, the mutational status does not really matter. It's probably more the size of the cells that will matter and how strong the field you can get than the genetics, at least at the current understanding. But in recurrent disease, EF11, tumor treating field had no significant effect. You could see some kind of efficacy with a little higher response rate, but it did not prolong overall survival. So that's one of the reasons I would use it up front early on. But here I don't think this makes much. I would not fight for this patient with tumor treating fields when you cannot cover everything with the field.
Yeah, thank you. Thank you so much. I truly appreciate your input.
Right. Okay. And just, I know we're at time here, but I'll just, my case is much more straightforward.
I hope so.
And just kind of brings up some of the points that you made earlier. This is a patient of both mine and Pilar, actually, a 63-year-old that presented a couple months ago with his first-time seizure. Very, very small enhancement in the right frontal lobe and with surrounding flare. So he had a aggressive resection, including the flare signal around the enhancement. And the biopsy came back as a GBM, IDH wild type, MGMT non-methylated. And here's the rest of the information. His Ki67 was extremely high at 30%. So we were ready to start the Stupp protocol. And 3.5 weeks later, on his MRI getting ready for radiation, he had a sizable recurrence that was larger, much larger than the initial tumor. And I've seen this now probably about 3 or 4 times in the last 5 years. And so the question at this point is and maybe I'll start with Dimitri, is what would you do in this situation? Would you go back in and re-resect, or is it cat out of the bag, and is it just time to move on quickly to adjuvant therapy?
Yeah, very interesting case. It would be, is the patient symptomatic now with this recurrence or not?
No, not symptomatic, doing well.
Yeah, it would be a tough sell because you would still have to wait a few weeks before starting therapy for things to heal up. I'm not sure what the right answer here is. one of my questions was related to the pathology. You said the H3K27 trimethyl immunostain. Did you mean K27 methine M? K27M shows focal loss, or?
No, I think E3 is a different subtype of that. But
Yeah. Okay. Yeah. It would be a tough sell to re-resect because what are you going to do differently? But you may have to. I'm not sure. Is the tumor MGMT methylated or not?
no,
No.
unmethylated.
Yeah. It would be tough. I guess you can try to re-resect, but try to expedite the therapy, subsequent therapy. Is there multifocal disease there? Is there?
It was congruency, and then these other two spots on the flare were not disease; they were just for due to age. So yeah, so we had a long discussion about this. He was, I think that the 3.5 weeks may have been actually 4 weeks because we were trying to get him into a clinical trial that we had here, and wasn't quite ready yet. And there was also some delay with him even starting the therapy because of within the next couple of days because of his insurance was changing. And so because of that, we talked to the patient, we ended up to proceed with another resection. And I'll get Dr. Stupp's opinion after this. So another resection, he did well. Again, very aggressive, got the flare signal, got the enhancement. And then he was then started on, he wasn't eligible at that point for any trials. He was then underwent their standard protocol and now presents 3 months later after this, after completing therapy with progression of disease. However, the perfusion scan shows it's not as impressive, but obviously with the extent of flare signal, there's additional mass effect and more enhancement. It's likely progression. So that just kind of brings me to the question of when you look at that, I want to know what Dr. Stupp's idea is early, again, surgery, would he recommend surgery in this case? And then this idea of survival impact on the initiation of chemoradiation after resection of tumor. Both myself and Andy Parsa and the rest of the UCSF group actually kind of published at the same time on this. And there was some findings that actually 4 to 6 weeks after surgery was kind of this best time point, and whether that's related to the steroid tapering or not. In this case, actually doesn't fall into that window because it progressed quickly. But weighing all those thoughts, I just want to see what Dr. Stupp's recommendations would be.
So challenging. Again, I think I would agree with what you have done. Mm-hmm. So I think with that regrowth, just in order to be able to deliver radiation, not have mass effect during radiation and so on, I think it made sense to re-resect. And then, as Dimitri has said, make sure that you already lined up that you can 10 days later start with radiation. Now, again, as a general, but we should not generalize this. It's usually not, those cases where we have to start radiation immediately and cannot wait 3 or 4 weeks. It's not going to make that. It's not, if you would have started already, it would not make a difference. When you want to get the resources for a new MRI machine, then we will say, for a new radiation machine, we will say that it matters. But when we looked at, and our Australian colleagues looked at, and even the big trials, we had a median of close to 4 weeks after. It didn't make a big difference. So it doesn't mean you have to wait, but if it would have been an MGMT methylated tumor, I may have advocated to start with chemo first.
Sure.
And sometimes, again, it's different in our big centers, but if I would be somewhere in the woods and it takes a while until I get the patient to radiation, have a methylated tumor, I think it's fair enough to start with temozolomide kind of neoadjuvant. We've looked at this at some point, whether there's any advantage of giving temozolomide in a neoadjuvant setting. It is not, because we don't get enough responses. It's not like breast cancer is chemosensitive and they get pathological complete response, or even head and neck tumors. So that's why it's not something to as a pattern, but for methylated ones, you can get it quite a bit, and we could talk on that. This is a poor prognosis patient. I don't know what this molecular histone change will help us with today's knowledge. Hopefully we learn more in the future. I think, but likely prognostically poor. But again, of the different cases, also things which have been discussed, also the cases Dimitris presented before, the high variability we have. But you said it's a high proliferation index. 30% is surely high, but I've seen 60, 70%, and I don't have had this kind of regrowth. Dimitris also showed the case which was over 70 and actually was doing reasonably well. I think this kind of super depressed attitude on people who are over 65 to not do anything is probably not the right approach. I think MGMT remains the best predictor for outcome, not on everybody, but we start learning the ones who are having a better outcome than predicted by MGMT methylated. These were exactly the ones which has this FGFR tax-3 fusion. So all the others actually, it works. And I think somebody was asking earlier, how we integrate all the molecular so we have better prediction where it's worth to invest or not, and also where we have to we should take more risk because there's nothing to lose. And what we didn't talk about, all the ones we should de-escalate and sometimes do less, especially on the lower grades, that we could fill another seminar on just that. When you have patients who live 10, 15, 20 years, and we do everything now, so nobody can complain that we haven't done enough, but it's the patient afterwards who has to carry the consequences. So even though it's the exception. So it's a fine balance.
But what would be your recommendation as the next step forward, for treatment for these patients? He progresses by the standard of care. My plan was to put him on Avastin and lomustine in combination, trying to give him a more powerful like a synergistic effect.
So what I think, fair enough. Again, do I know? I can only give you my gestalt and my way of thinking. Mm-hmm. On data, I would say, don't do less. But I have had those patients as well. So sometimes I go back to Hurlinger's paper in Germany, who combined temozolomide and lomustine. Now he did it on MGMT methylated ones. And I'm not sure that there was a true benefit out of the trial. What he has been hiding in the supplement is the true intent to treat, not the modified intent to treat, which was no difference. But what he has clearly shown is overall it can be done safely. So I've been using that on an unmethylated when I have nothing to lose, and I feel rather than giving temozolomide first and then lomustine, I throw everything in upfront, and then I'm more restrictive what I do at recurrence. You've been mentioning bevacizumab repeatedly. I would use bevacizumab generously by putting the indication, as long as I have reassured myself with Michael that he doesn't want to do more surgery and the wound is fine. But I never give 10 milligrams per kg. I give 3 to 5 as mentioned. It's much better than steroid. And I think there was one of the questions in the chat asked, bevacizumab and its cost and so on, and steroid. I think we should have developed bevacizumab instead of steroids at the low dose, at the frequency needed, and it's much less toxic, less infection and so on. So that's when I would use it. Now, again, if you have a patient you want to get on clinical trials and so on, it's a different story. But here we talk now not as researchers, but we talk as physicians when I have to come up outside the clinical trial consideration, what is the best for my patient to gain quality time.
Great.
Mike, can I ask a question?
Yeah.
Did the pathology, did those markers change in your second surgery? That focal loss of the H3K27 trimethyl is very interesting. That's the loss, that's a chromatin modification that you see in K27M, the methionine mutation. It just poisons the K27 trimethylation. And in a perfect world, I think this tumor is probably treatable with inhibitors of the activating chromatin marks, but that's a long story. I was just curious whether there was any change between the original and the second path, but there wasn't.
Yeah, no, there wasn't. There wasn't. But thank you.
Good point. I think looking also, don't believe all the molecular markers, double check who has done it. I always write which lab has attested X and Y. I think, and then looking, that's where the field is going. I took it for the FGFR tag, but you could look on NTRK. There are other compounds out there, some proven to be effective, some is less proven, but at least the mechanism.
Right. Well, I just want to say thank you again to everybody. We're over time, and I don't want to keep anybody too late. I know everybody's got many things to do, but I really appreciate Dr. Stupp. Everything that you said tonight was amazing. I'm going to make sure all the residents and fellows watch this over and over again as many times as we can get them. And Demetrius and Pilar, also thank you for the great cases and insight. Please, everybody stay safe, and we'll see you here next week.
Thank you very much. Pleasure.
Thank you very much.
For having me.
Dr.
Bye-bye.
Stupp, nice to meet you.
Bye, Pilar. Bye. Thanks. I will respond to the chat separately.